Hi students, today I am here with another type of inheritance pattern. We already discussed the inheritance pattern in pea plant and snapdragon. In pea plant it is complete dominance, whereas in snapdragon it is incomplete dominance. Today we can discuss another type that is known as the codominance. The best example for studying codominance is human blood group, ABO blood group system. According to that system, there are 4 types of blood groups. Which are they? A group, B group, AB group and O group. The plasma membrane of the RBC contain a sugar polymer. These protrude out of the plasma membrane, and the kind of the sugar polymer synthesized is controlled by a gene known as the I gene. It controls which type of sugar polymer is going to synthesize. And there are 3 alleles of this gene I. Normally, a gene possesses a pair of alleles, but here in the case of I gene, there are 3 alleles. These alleles are IA, IB and small I. This IA that controls one type of sugar polymer, IB controls another type of sugar polymer, whereas this small I is not able to produce any kind of sugar polymer. Okay, you know the classification of A, B, AB and O. It is based on the presence or absence of certain antigens or antibodies. And if this IA gene appears in homozygous condition, then that blood group is known as A group. Because it produces a particular type of sugar polymer, this blood group is known as the A group. Okay, and if the allele B, IB, it occurs in homozygous condition, it produces another type of sugar polymer, and this blood group is known as the B group. And if this small I, it occurs in homozygous condition, there is no sugar polymer, and that results in O group. Then think about if both IA and IB come together, IA and IB, which one will express there? Here, in the case of this blood group, both the genes express together, and that results in AB group. Because this IA synthesizes one type of sugar polymer, IB synthesizes another type of sugar polymers. That means both these express together, and that type of inheritance is known as codominance, expressed together. Okay, that is codominance. And here I wrote IA for A group. This is homozygous condition. Okay, and if it is in heterozygotic condition, how can we write? IA small I, because it is recessive. So IAI, it also results in the A group. And if it is IB small I, then it also become B group. So these are the different genotypes of different blood groups in human beings. The same gene can control or produce more than one effect. For example, if capital B, this gene is responsible for the starch production in pea plant, pea seed. Okay, if it appears in the homozygous condition, capital B, this gene results in the production of a round seed because more starch content is produced. If the gene is in homozygous recessive condition, less amount of starch is synthesized and the seeds become wrinkled. And if the condition is heterozygous, then medium amount of starch will be synthesized. So we can say it is also an example of incomplete dominance in the phenotypic expression. We can say like that. Okay. Now let us study a typical Mendelian dihybrid cross. In dihybrid cross, two pairs of characters were selected. Here I wrote one example, tall axial and dwarf terminal. In the F1, all the progenies obtained were tall axial. But when it is self-pollinated, he got tall axial, tall terminal, dwarf axial and dwarf terminal progenies in the ratio 9:3:3:1. Okay, the cross you already studied in class 10 also. So here the maximum number, that is 9. This is the dominant parental character. The least number, that is 1, that is the recessive parental character. Then what about the others? It is the new combination, non-parental combinations. Okay, tall terminal, dwarf axial. So after conducting this experiment, Mendel formulated his third law of inheritance, that is known as law of independent assortment. According to this law, two pairs of characters are combined. The segregation of one pair of characters is independent of the other pair of characters. Mendel published his works in the year 1865, but it remained unrecognized till 1900 because communication was not easy at that time. Second thing, Mendel's concepts of characters were controlled by discrete stable units called factors, the allelic pairs which do not blend with each other. These things were new and the biologists at that time, they could not accept this. Moreover, Mendel failed to provide any physical proof for the existence of such factors. Another thing, Mendel used mathematics to explain these biological phenomena. That also could not accepted by other biologists. But later in 1900, three other scientists, Correns, Dihybrid and Sharma, rediscovered Mendelian works. And at that time microscopy were developed. So they observed the chromosomal movement during cell division also. Later, Sutton and Boveri, they find out the chromosomal movement. It is similar to that of the genes or factors. Chromosomes also occur in pairs. Genes also occur in pairs. And these genes, they are placed in the chromosome in specific positions. These positions are known as the loci. And Sutton and Boveri argued that the pairing and the segregation of chromosomes would lead to the segregation of the traits that carry. Because the characters are controlled by genes, these are placed on the chromosomes. So when the chromosomes segregate, that particular character will also segregate. And they combined the results of the Mendelian principles with what they observed on chromosomal movement, chromosomal behavior. They combined both these things together, and that is known as the chromosomal theory of inheritance. Okay, and T.H. Morgan, Thomas Hunt Morgan, experimentally verified the chromosomal theory of inheritance by performing certain experiments in Drosophila melanogaster. It is the common fruit fly. He selected this Drosophila because it can easily cultivate in any synthetic medium in the laboratory. Second thing, in a single mating, it can produce large number of progenies. Third thing, the male and female Drosophila can easily identified. There is a marked differences. So it is very easy to identify which is male and which is female. Moreover, it shows many physical hereditary characters. Okay, so many variations it shows. So Morgan selected Drosophila for his experiments. And after the experiments with the Drosophila, he understood the dihybrid ratio, the F2 ratio is significantly deviated from Mendelian dihybrid ratio. Why this variation happened? That we can study in the next class. Thank you.